So what we looked at now is between complex 1, 2, 3 and 4 is this free electron, or 2 in the case of complex 1, 2 electrons on complex 2 that happen to be carried by CoQ10 to complex 3 and then by cytochrome C to complex 4. So what we know is, which we introduced in the last module, if we add those electrons univalently to oxygen, we get superoxide if we add 1. If we put 2 electrons on univalent, we get hydrogen peroxide. If we add 3, we get hydroxyl radical. And if we add 4, we get water and molecular oxygen again. And we know from this pathway, the dark blue lines here are the normal pathways of eliminating these reactive oxygen species, or univalently added. We know that we produce these reactive oxygen species, superoxide, in 5%, maybe less, of normal mitochondrial oxidation processes. So we're producing a certain amount all the time in the mitochondria. When we've got an infection and we have the respiratory burst, when we got phase 1 detoxification through cytochrome P450 enzymes, and in the situation with hypoxia, or rarely hyperoxia if we're in a diving bell tank. So the normal pathway is mediated by these enzymes, superoxide dismutase, catalase, glutathione peroxidase, etc. On this side, we got the reactive nitrogen species from arginine to nitric oxide by inducible nitric oxide synthase here. Then what we did yesterday is we filled in, last module, we filled in the underlying enzymes here are the heme-dependent ones. So heme we're going to be looking at shortly, how we make heme in the pathway of making hemoglobin. So these are all iron-dependent enzymes. So inducible nitric oxide synthase, cytochrome P450, the cytochrome C, the myeloperoxidase, NADH, peroxidase, other peroxidase, and catalase. These are all heme-dependent enzymes. So if we can't make heme properly, we can't make these enzymes work for the immune system. And as we learned in the last module, we make these free radicals, or reactive oxygen species, to kill microbes by the innate immune system. So gram-positives here with the hypochlorite, gram-negatives with the nitric oxide. Hydrogen peroxide kills parasites. Peroxynitrite kills really stubborn viruses and fungi and parasites if they're really stuck. And hydroxyl radical, probably the same, particularly with mycoplasma chronic infections. So we've talked about how to challenge for energy. A weak muscle strengthens to ATP, strong muscle weakens to ADP. For glycolysis, challenge strengthening against pyruvate, and weakening in this case to lactic acid, particularly if they're a sports person. For Krebs cycle, strengthen against NADH or FADH2, challenge the individuals for Krebs cycle intermediates. For the electron transport, in the pain diagnostic kits, you've actually got complex 1, 2, 3 and 4, but complex 1 is NADH, complex 2 is FADH2, you've got a complex 3 there, and complex 4 is basically cytochrome C oxidase enzyme. So let's have a look at gluconeogenesis. Genesis means creation, and therefore this is the creation of glucose. And this traditionally is done by the burning of amino acids from proteins. So when we ingest and digest proteins, we chop them up into single amino acids, and these amino acids go into the pool. In other words, in the bloodstream, we have a certain number of amino acids. And these amino acids then can be burnt as fuel. So we see here hydroxyproline, serine, cysteine, threonine, glycine, tryptophan, and alanine can go in at this point of pyruvic acid here. So we see pyruvic acid here can go through on the one-way trip into making acetyl-CoA. So in the Krebs cycle itself, we've got fumarate here, and we've got tyrosine and phenylalanine that can go in at this point, and histidine, proline, glutamine, and arginine can go in into alpha-ketoglutarate. So what happens is I've now put a turquoisey blue colour there, so you notice they can go either way there, but at this point they can only go forward. So what happens is the amino acids convert to fumarate or malate and then go back up the blue cycle. Let's go back here. And it goes up the blue here, this pathway, up and out, and is formed to glucose, and then it comes back down again. So it's a long-winded pathway, gluconeogenesis, of converting amino acids basically into glucose, which then goes through the pathway of the liver and comes back down again. So we can burn amino acids, but it's inexpensive and it's a long pathway of doing it. Whereas obviously glucose is the first form of energy, and then the better form is then glycogen secondly, and thirdly would be fats. So burning of fats or beta oxidation is the preferred way here. So when we get to acetyl-CoA, if we don't spin the Krebs cycle fast enough, because we've got a sluggish thyroid, we get a build-up of acetyl-CoA. Okay, you can understand that because you're not burning your substrate. This will shunt it along this way to malonyl-CoA, which is a biotin-dependent enzyme, which then goes on to make fat. So the fat deposits eventually usually in even-numbered fatty acids, saturated fats, 16, 18, 20, 22. Palmitic being the soft, buttery-like fat. Carbon 18 is stearic, which is more harder. This is the candle wax type of fat. Arachidic, not arachidonic. Arachidic is carbon 20, which is more solid. And bahenic is 22, which is one of the highest saturated fatty acids that the body uses. It's a real firm one. And what makes them firmer is the melting point of each one. So they get higher melting points. This makes them harder to shift. So the higher the fatty acid, the bahenic and the arachidic are much harder to shift to burn than the stearic, and the stearic is harder than the palmitic. So how we normally store our fats is palmitic, which is a softer, buttery one. And if we put 3 palmitics together, we get triglycerides, or triacylglycerol here. So we form them in threes. So tripalmitate would be 3 palmitics. Often we have a stearate and 2 palmitics, or any combination there. So you get varieties in the body of things like lipomas. You know, when you get a lipoma, a fatty lump, which just doesn't shift at all. These are usually arachidic or even bahenic. So you've almost got to boil the person to get the temperature up to start to melt those. Very difficult things to shift. So we store the fat as triacylglycerols or triglycerides, okay? We then have to break that down when the time comes, when we want to break the fat down and use as energy. And to do that, we need lipase. Now, this is the same lipase that's secreted in the gut, is actually secreted inside the body, inside the mitochondria here, the fat cells. And what it does is it splits the triacylglyceride or the triglyceride into 3 molecules of palmitic acid or whatever the fatty acid is. So this is hormone sensitive lipase, this is called. So it's a lipase which is directed by certain hormones. Now the main hormone that regulates the breaking down of fat is our thyroxine. So thyroxine itself regulates the speed of the metabolism, and therefore when we run out of fuel, it stimulates lipase to break fat down. So this is why generally if you've got a hyperthyroid, in other words an overactive thyroid, you lose weight, you lose fat. And if you've got an underactive thyroid, you gain fat. Okay, so it's all to do with the making and the burning of fat. That's the main one. The second one which does this is adrenaline. So adrenaline is when it comes in when we're under stress. So we burn fat when we're under stress because we want to get out of that stressful situation physically and run like hell. Okay, so we need lots of energy in there. So if you're under stress for a considerable period of time, mental stress or whatever, and of course remember that all exercise is a stress to the body, so we stimulate our adrenaline when we exercise, which helps us to burn fat. But if we're under mental stress, we'll do the same thing. The high level of adrenaline will make you burn fat and you lose weight. So the fastest way to lose weight is to get a divorce and go on a diet. Okay, divorce, diet, and then the financial settlement. And that'll knock the pounds off you beautifully. Okay, so your adrenaline is right up by that time, and you soon lose a lot of weight. So palmitic acid, the hormone lipase is adrenaline, which is for stressful times, and our thyroid hormones for normal. So we split the triacylglycerol triglyceride into 3 molecules. This then goes across the outer to the inner membrane of the mitochondria. So this is carried by palmitoyl-CoA, which is just recycled. So the CoA, remember, is B5, but to get to this, we need acyl-CoA, we need B5 and magnesium to start to burn the fat. So again, our Bs are very important here as far as burning fat. And this recycling of the palmitic acid from the outer to the inner in here to burn it is called the carnitine shuffle. Sounds like a dance, doesn't it? The carnitine shuffle. This is where the amino acid carnitine, which is made from methionine and lysine, carnitine shuffle, carries the palmitic acid across into the furnace, if you like, and eventually through beta oxidation, as it's called, to make acetyl-CoA, which is the fuel for the Krebs cycle. So that's how we burn fat. So to burn fat, we need to split our fat molecules up, and then we need to carry them across into the mitochondria with the carnitine shuffle. Now, carnitine itself can be taken as a supplement. The body shouldn't normally make it, as I say, from methionine and lysine, and it needs a lot of vitamin C to be able to do that. But you can buy carnitine supplements, and possibly better than carnitine is acetyl-carnitine. This seems to get in there with the acetyl group very well, and the body can use the acetyl group to go on to make acetyl-CoA and acetylcholine is a good acetyl donor. So acetyl-carnitine is also the fuel that the sperm use with their tails to get up the various tubes. So carnitine shuffle, and when we go through this final process called beta oxidation, where carbon 16 goes to 14 to 12 until it gets right down to acetyl-CoA, which is carbon 2. So the palmitoyl is transported into the mitochondria using the carnitine shuffle here. So the carnitine is basically just the carrier. It's the sort of carrier molecule of the fat across the 2 membranes, the outer and the inner membranes there. Here's the beta oxidation pathway itself. So we've got here to our palmitoyl, carbon 16, then we go to 14, 12, and each one of these needs FAD, or the active form of B2, and NAD, which is the form of B5, and CoA, which is the form of B5. So we've got B2, B3, and B5. So again, all our Bs are necessary to burn fats. So then we end up with acetyl-CoA here, lots of molecules of acetyl-CoA, as you can see here, from burning fat. So fats is the most efficient way of creating energy, but sometimes the hardest way to switch into burning. So you could switch into burning if you just don't eat glucose. Okay, and this is what the modern ketogenic diet approach is. Don't touch glucose, which is going to force you into burning proteins. So you eat your high protein diet and fat, so you get into this ketosis regime. I think the jury's out on it. I don't want to go down that route because there's some people who found fantastic, but they look awful and they stink with their breath. And you think, you know, what penalty is the price that they're burning fat? If they feel good, nobody else does around them. So whether it's right, I think to me a varied diet is the most important thing at the end of the day. So that's the beta oxidation. So let's just have a look when we break fats down, or when we break proteins down, is this breaking down of amino acids, which are normal alpha amino acids we call them. When we deaminate them, in other words, we take away the amine part of the molecule here, we end up with ammonia. So high protein diets lead towards high doses of ammonia in the body system. And ammonia has to be got rid of to make urea in humans. In animals, they don't bother about urea, they just excrete ammonia, which is why their urine smells. Okay? Whereas humans don't excrete ammonia, we excrete urea. So we can produce ammonia from the breakdown of proteins here through transamination. And all transamination requires B6, or pyridoxal 5-phosphate, to be efficient with it. So here we see B6 coming in to the usual ones here. So we can also produce ammonia from enteric bacteria and parasites. So parasites will give off ammonia, and enteric bacteria, usually the gram-negatives, will also produce ammonia in the gut. So because a person weakens to ammonia, you don't know that it's just transamination. What we do know is we've got to get rid of ammonia because it's a very toxic substance to the brain and to the kidneys. So this is the urea cycle. So here's the carbon dioxide and ammonia coming together. And this first pathway, the carbamoyl phosphate synthase enzyme here, which I put in again the chromosome number and the wavelength of each of these, is biotin dependent. So now we see where biotin comes in as an important part of the metabolism or the breaking down of ammonia. So carbamoyl phosphate goes to citrulline, goes to arginosuccinate, goes to arginine, and then with arginase enzyme here goes to making urea. So you see urea is basically two molecules of ammonia joined together with a carbon and an oxygen there. And then when you remove that, you're left with this amino acid derivative called ornithine, which then just recycles back again to citrulline. So as the ammonia comes in, it cycles round rather like the Krebs cycle. Now when we get to arginine here, we can convert arginine to citrulline by the nitric oxide synthase pathway, which we saw that nitric oxide was a reactive oxygen species, and that's how we produce it, the NO or nitric oxide. So that's the pathway, arginine to citrulline. This all occurs in the liver, by the way. Okay. So we produce urea in the liver, and this arginase enzyme has often got polymorphisms in it, which means we don't get rid of the ammonia. So we tend to recycle it, and people get high levels then of ammonia in the body. And what that will do is give them the EPGBs in the middle of the night and when they wake up. And they really get nasty feelings of real fears and dreads and doom and gloom. So if you've got a person who weakens to ammonia, you can tell them, do you get these feelings of doom and gloom and dreads and things? And they say, well, yeah, how do you know? And you say, well, because you've got high ammonia, and because it's produced in the liver, it's going to be 2 to 4 o'clock in the morning, and they wake up and they have these awful feelings. And then during the day when they're up and about, well, they leave them because the exercise and things and moving around tends to break it down and help, and they drink water and so they wee it out better and so on. And then the next night it comes back again, and they can end up on severe antidepressant drugs and all sorts of things. And all it is a change of the chemistry, which gives these horrible negative feelings to put them on, first of all, ornithine, because that absorbs then the ammonia, and then have a look on the chart and say, this enzyme is manganese dependent, zinc and vanadium. So those three elements there are the cofactors for the arginase enzyme to get rid of ammonia. So you can pick again this pathway apart, but most of the time it's arginase. Put them on ornithine, which is like the blotting paper, absorbs the ammonia, but it's not the answer to the problem, but it can save a lot of the distress that they go through. But look for how to open the pathway up each time. So it could be biotin that they need here, could be manganese here. We've got P5P, all our friends, the usual magnesium, P5P, all coming in.